more minutes talking about another approach to generate T cell responses that does not involve using a therapeutic vaccine, but instead the idea is go directly to T cells and manufacture and infuse T cells that have the right specificity into patients. Again, many of you may be familiar with this general approach. CAR T cells have been developed over the last few years that have demonstrated, really just incredible effectiveness in a few types of cancer. And the basic premise for those of you who are unfamiliar with them is to take T cells from a patient, to genetically modify them so they specifically recognize cancer antigens, and then activate these cells very effectively to make them good at killing those cancer cells. These are then infused into patients, and here we're just looking at a couple images from one of the early trials. And everywhere you're seeing the black in the main part of the body, and the bone is where these cancer cells are existing. And after the CAR T cell therapy, you can see a really incredible clearing of the cancer throughout most of the body. This has led to over 80% overall remission in a couple of different types of blood cancer with some of these CAR T cells, which was just unprecedented finding. And this is similar to the checkpoint blockade, led to a lot of excitement, many ongoing clinical trials. As of about a year ago, there was over 750 T cell based therapies under development for cancer alone. So a really exciting avenue of research for cancer treatment. And the process looks something like this, where isolate T cells from the blood of patients, typically isolate about 100,000 or a million of these. These then get activated in the lab. They get genetically modified, and then these cells have to be expanded and then ultimately reinfused back into a patient. The whole process takes about 3 weeks. About half of it is the expansion step. So the timing of this can be an issue because it takes so long. And then the quality of the T cells is actually poorly controlled with the current approaches, where we don't have a lot of ability to manipulate basically the type of T cells and the function of those T cells. So while CAR T cell therapies are really successful in certain types of cancer, they are not in others. And overall, there's a variety of challenges to making these cells, including their cost, having control over the type of T cell and the response you will get, and then simply the time. It takes so long to manufacture these cells that, unfortunately, a significant fraction of the patients die before they can get their own cells back to be treated. Now, if one looks at the way these cells are currently expanded, there's some real limitations to these systems. The biological approach is the one shown at the bottom, where after you isolate the T cells, you also isolate antigen presenting cells from the same patient, and you use those cells to stimulate the T cells in the lab to generate all these therapeutic cells. Now that requires that you isolate not just the T cells, but also dendritic cells. If the patients have an underlying disease, you may have genetics issues in terms of the ability of those cells to really promote robust T cell expansion. So that becomes very cumbersome. So the strategy that is used widely is to use an artificial dendritic cell. And the most common is something called dynabeads, which are polystyrene beads to which the cues that dendritic cells normally use to stimulate T cells are conjugated to the polystyrene beads. The polystyrene beads act as an artificial cell. The challenges from that approach, including the fact you have to remove the particles after you make the cells, and you're probably not getting an optimal activation of the T cells because the way these cues are presented is not the normal way a cell would present these cues and engage with the T cells. So what Alex Chung here in the lower left, and then David Zhang, a current PhD student in the lab, proposed a couple of years ago is that one could make a better artificial antigen presenting cell or artificial dendritic cell using some of the particle-based systems that I described to you earlier, in particular the mesoporous silica. And the basic strategy they chose to pursue is shown here, that after they fabricate the particles, the first thing they do is absorb the cytokines of interest so that the particles can then eventually release these over time, mimicking the way the dendritic cells release agents to T cells. They then coat these particles with liposomes to create a supported lipid bilayer that would mimic the kind of cell membrane of a dendritic cell that the T cell would normally interact with. They attach the appropriate cues to the supported lipid bilayer, so now they will be presented in a much more natural way, And once this system has been created, you could simply mix it together with PBMCs or the T cells to promote their expansion. Now, this is what the particles look like after they've been coated with the liposomes. So you can see you get an even coating over the particles. We do have a supported lipid bilayer that is fluid, as indicated by this FRAP test, where we photo bleach the center region, and you can see we get a return to fluorescence, indicating that the lipids in the surrounding region readily diffuse over and replace those that have been bleached out. You can get a sustained release of cytokines, mimicking how cells would provide a sustained release. In this case, you're looking at about 5 to 6 day release of IL-2, which is a really important cytokine released by these cells. Now, unlike the natural cell, what we can do here, though, is we can precisely control the amount of the cues that we incorporate into these artificial cells to potentially alter the way that they interact with the T cells and stimulate the T cells. And I'll come back to this later. And this is just what these particles in green look like once you've been mixing them with T cells in red. And you see the T cells readily engage with the particles and bind to those particles. Now, to induce a polyclonal T cell expansion, what we typically do is bind antibodies to the supported lipid bilayer that will bind CD28 and the T cell receptor on the T cells. And if we then expose these to primary human T cells and look to see how readily we can expand these cells, you can appreciate the APCMS leads to about a 4,000-fold expansion of these cells over a course of 14 days, while the Dynabeads, which is the industry standard, lead to about a 250-fold expansion, and the mock don't expand at all, as you would expect. So it looks like we're able to get a much more rapid expansion of the T cells with this system as compared to the way they are typically expanded. Now, if the cells ended up being exhausted, which would mean that they would be poorly functional, then it wouldn't do you a lot of good to have many cells. So we've also looked a variety of ways at the function, which I'm not showing you, and the exhaustion, which you are looking at here on the right. And you can appreciate when we quantify the percentage of the cells that are PD-1 LAG3 positive, you can see that at the end of the 14-day expansion, we have very similar percentage of the cells are exhibiting these markers of exhaustion as one sees with Dynabeads. So it looks like we can get greater expansion without compromising function when we do a polyclonal T cell expansion. Now we can also use this system to do an antigen-specific expansion. And in this case, instead of having an antibody that generically binds to the T cell receptor, we instead use an MHC molecule that presents the antigen of interest. And to demonstrate the potential of this approach, I'll just show you some data here where we isolate T cells from patients. We screen for expression of HLA-A2 in prior exposure to the Epstein-Barr virus. So we actually know that they have T cells that would respond to a particular antigen. We then fabricate the APCMS system with EBV-related peptides, and we look to see whether we can expand these cells. And what we find is that if we look at the expansion of the T cells, in this case, the orange indicates we have the appropriate peptide to get expansion of those specific T cells, while the black clear symbols means we have the other irrelevant peptide. And what you can see on the left when we're trying to generate CLG-specific T cells, with the APCMS, we get a really significant expansion in these cells. And what you'll notice with the monocyte-derived dendritic cells, the MODCs, we get a much more modest expansion. So it looks like the system can dramatically outperform the natural system, the autologous dendritic cells, in terms of expanding these cells. And the graph on the right just shows you the same kind of result, but now looking at expansion of a different T cell population in those cells. So we can dramatically increase the expansion of these cells. And as I mentioned earlier, since we can now actually have control over the level of stimulation provided by the particles, we can actually tune the T cell products. So this just represents some data on the right where we've taken a bunch of formulations that are not identified specifically here, but we vary the amount of particles, the amount of stimulatory molecules on each particle, and we've then cultured them with the human T cells, and we've looked at our ability to control the CD4 to CD8 ratio, which is considered to be an important metric for their effectiveness in vivo. And you can see we can readily dial in various CD4 to CD8 ratios. And furthermore, on the bottom, what you're looking at is that depending on the formulation, we can have a significant impact on the ratio of effector cells, effector memory, central memory, stem cell memory, simply by altering the formulation of our system. So we can now start to tune the types of T cell products that arise with this system. To demonstrate whether this actually enables us to have a real functional impact back in the body, we've gone back to the same model that was used to validate the original CAR T therapies. In this case, using human cancer cells that are introduced into an immunocompromised mouse. We introduce these cells, we give them basically a head start, and then we introduce T cells, the CAR T cells, to try to control tumor growth. And the CAR T cells we're using here is actually the same system that was originally developed by Novartis. So they're second generation 19BBZ CAR T cells. And we are now comparing the effectiveness of APCMS to the Dynabead expanded cells. If first we just look at the ability of the two systems to expand these CAR T cells, we can see, similar to the primary human T cell data I showed you a minute ago, when we use the APCMS, we get a significantly accelerated expansion of the cells with our system as compared to the Dynabeads. So we can actually generate a lot more cells more quickly. And a really nice feature that I did not mention before about this mesoporous silica is it dissolves over time in the cell culture media, and by about 7 to 8 days is undetectable. So we don't, at the end of the manufacturing, have to remove it because it simply is gone, it's degraded away. And now if we take the same number of expanded cells, either from the Dynabead or the APCMS, and we infuse them in these animals, and we look to see how well they control the tumor or the cancer cell growth, you can see in the middle graph, we're just looking at the total flux as an indication of basically the growth of the cancer cells. And if there's MOC, which is basically untreated, you're seeing more or less an exponential growth. If we infuse either the Dynabead or the APCMS expanded T cells, we get a significant knockdown of those cancer cells. And by the normalized flux, it looks fairly similar. If we look at the percent survival, again, the APCMS expanded cells on a per cell basis are at least as effective as the Dynabead expanded cells and actually give us a statistically significant improvement in terms of survival in this model. So it looks like we can generate cells with a great flexibility, multiply those cells much more rapidly, which presumably could allow patients to be treated more quickly and actually have a more functional cell at the end of the day. So we think this system has a lot of potential for T cell expansion in the format as I just described to you and is indicated in this schematic. However, we really think it's probably going to be a much more broadly useful platform which we can incorporate a variety of other types of signals besides those that I've already described to you, and potentially be useful for the expansion of a variety of other cell types besides T cells, including various immune cells and perhaps hematopoietic stem cells as well. So this technology, I should mention, was licensed to a startup called Imulus, who are currently working to commercialize this technology. So kind of wrap up, a couple last comments. Hopefully I've been able to describe to you how we're using biomaterials to create what I would describe as cell factories, either inside or outside the body. The basic premise here is we want to control in time and space how the cells act with immunomodulatory agents. And we do this by bringing the cells to the device so they have an intimate contact, and then we unload these cues to the cells. We think that this creates really a platform or several platforms for cancer therapies, and I didn't have time to describe today, but this also provides a strategy to promote regeneration in a number of settings as well. And building on all the work I've been describing today, we've recently been funded by the National Cancer Institute and the National Institute of Aging to establish a center to improve immunotherapy called an I3 Center. This is part of the Cancer Moonshot Initiative, and we're developing biomaterials to promote T-cell immunity, including the types of APC mimetic systems I described in the last part of my talk, therapeutic cancer vaccines, as I described in the first part of my talk, and then also systems to promote differentiation of T cells from progenitors to enhance overall T cell reconstitution. So I'll stop here, and I'd be happy if there's some time left to take any questions.
Thank you, Professor Mooney, for the excellent talk. And if anyone has any questions, you can please enter those into the questions box. And so we had one question that came in during the talk. And the question was, how does the implant vaccine affect the T regulatory cells?
Yeah, so for those who aren't so familiar with the space, there's a cell that you can think of almost being the polar opposite of the cytotoxic T cell that I've been talking about today, the T regulatory cell, whose job it is to dampen immune responses. And so if we increase the number of T regulatory cells in parallel with the cytotoxic T lymphocytes, we probably won't accomplish very much. So what we found is that the impact on the T regs is highly dependent on how you formulate your vaccine. If you do not have the right types of adjuvants to activate the dendritic cells, you can actually lead to an increase in T regulatory cells. But if you formulate with the appropriate adjuvants, so you activate the dendritic cells effectively, you can get a significant increase in the CTL response without increasing the regulatory T response. And the net result then is you get a significant increase in the ratio of the cytotoxic T lymphocytes to regulatory T cells, which is what you desire in the context of cancer.
So one question I had was, as you're trying to develop these therapies for engaging the immune system, it seems like in many cases you need to use an animal model. And I was wondering if you could comment on, especially in some of the earlier studies, how much in vitro culture was used versus how much was done in the animal platforms?
Yeah, so we will always do some work in culture to validate that the system works broadly in terms of, do we release the agents of interest? Are they still bioactive? But it's very difficult to recapitulate the kind of interactions that require multiple cells engaging with each other in vitro, oftentimes. So the field has historically heavily dependent on animal models. Now, the negative of that is then you have an animal immune system, which is not the same as a human immune system. So there's a lot of interest in developing better in vitro model systems, organ on a chip types of systems that would enable you to use human biology in a setting that would be more reasonable and more consistent with in vivo biology. And like one of our projects in collaboration with Jennifer Lewis and Steve Hody is to develop basically vascularized tumor and vaccine models that we can do the kinds of studies you're talking about, but do them in vitro with human biology. But at this point, those systems across the board are being developed, but are certainly not ready to replace animal studies.
Yeah, okay. So going to some questions from the audience, there's a question, can you discuss the role of macrophages in the response?
Okay, yeah, so unfortunately I can't all that much. We've largely ignored the macrophages, even though they are clearly a really important antigen presenting cell. So we have a fair number of macrophages that are also concentrated at our vaccination sites typically. We've not done much work to really try to break out what their contribution is in the kinds of immune responses I've been showing you. We know that the dendritic cells are required because if we go to transgenics where we eliminate certain dendritic cell subsets, for example, the plasmacytoid dendritic cells or the cross-presenting dendritic cells, which there are knockouts available, we lose effectiveness. So macrophages are undoubtedly playing some role, but they are not the main player and are not able to generate the kind of responses that we see in the lack of dendritic cells.
And then another question we have is, how do you separate the T cells from biomaterials after expansion and stimulation?
Yeah, so with the system we're developing, the nice feature is that those particles will just naturally dissolve. So we don't actually have to do a separation at the end. When one uses something like Dynabeads, you do have to do a physical separation. So they have a different density, so one can typically do a separation based on density. So for example, centrifugation to get the isolation, which can be certainly and is routinely done, but just adds a little bit of complexity.
Yeah. Okay. So our next question is, how is the self-assembly maintained inside the body, and how is that enabled to reach the tumor target?
Yeah, so maybe I'll address the second part to this. So the idea is not that we're trying to get these materials or particles to traffic to the tumor. we typically are actually vaccinating at a site distant to the tumor, um, and and that may actually be advantageous in that there's probably less influence of the tumor on the immune cells and the resulting immune response. However, having said that, in a number of studies that I didn't show today, we've also been vaccinating directly at the tumor site to try to take advantage of all the antigen that's there. And that can also be an effective approach, though it's more challenging in that then you actually have to get access to a tumor site. For something like melanoma, can be straightforward. For other tumors, can be very complex. But in any case, we're typically placing the vaccine by injection where we want it to be. We're not trying to get it to traffic. Instead of the cells traffic to it, instead of trying to get the vaccine to traffic to the cells. And what was the first part of the question that I didn't address yet?
How is the self-assembly maintained in the body?
Oh, yes. Okay. Yep. Okay. Yeah. So I would be very careful. If I said the word self-assembly, I apologize. I shouldn't call the MSRs self-assembling. I would call them kind of maybe self-organizing. It might be as far as I'd want to go. There's no specific chemical interactions that's causing an assembly process of those particles. More or less, they're present in a dilute, relatively dilute manner in the saline. And so they typically are not contacting each other to any significant extent. As the saline dissipates, they will become more and more concentrated, and they will then collapse on each other to form a porous three-dimensional structure. But I wouldn't call that a self-assembly process as we typically in the materials world use the term self-assembly.
Okay. So another question we have from the audience is, do the biomaterials used in the whole vaccine system have any adjuvant effect?
Yeah, so they certainly can. Actually, our first generation system, we used the WDVax is fabricated from polylactide coglycolide, and we specifically chose that because it's known to induce a mild inflammatory response. So we consider that would probably be a positive here. And the mesoporous silica is actually quite a bit more inflammatory than the polylactide coglycolide. And we think that actually is an important element of it because as we change the surface chemistry, we can change kind of the baseline inflammatory response. And we do see a difference in the resulting adaptive response depending on how inflammatory the particles are. And we can go the other way. We can use systems that are really non-inflammatory like alginate or other hydrogels. And there, if we don't include an adjuvant, we really get no effect at all. And actually, they seem to be induce tolerizing effects as if we don't generate or don't include appropriate adjuvants. So yeah, so the material chemistry is an important consideration.
That's a very interesting point. I guess to build off that one step further is in the work you were presenting towards the end, I know you were saying you were coating liposomes onto the MSRs. I guess could you maybe talk about a little bit further about the role of the liposome coating?
Yeah, so the role there is we have we're presenting the cues that will engage the T cell receptors, and we're now doing it from what we think is actually represents something quite similar to a cell membrane. And when a T cells engage with these agents, they will typically concentrate their receptors and create something called a synapse, a particular structure. So when you present these agents from like a rigid surface, like a polystyrene bead, the cells have very limited ability to rearrange these agents and to form a synapse with the particle. But now when these are presented from a fluid membrane that resembles a cell membrane, as the T cell grabs these and begins to pull, they can actually remodel these ligands and form a synapse with the particle that should be a more natural signal to the cells as compared to when they engage with the same signals in an immobilized state.
Yeah, that's a really cool way to leverage that. And I guess to follow on one step further, do you think those liposome coatings might be used in other applications in regenerative medicine with the scaffold coating?
Yeah, I mean, I think potentially it could. We've not explored that yet, but I think it's something that could be, there's a variety of settings where having an artificial cell may potentially be useful. And I think that can be an interesting approach to try to get something a little closer to the natural signaling interactions that you see.
Yeah, that's a very cool idea there. So another question we have from the audience is, what are the risks of generating autoimmunity using a patient's tumor biopsy for cancer antigens?
Yeah, so that's always something that is a potential concern. In our first generation device, that's what we're using for Amgen is a lysate. Um, at this point, we've not seen that occur. Clinically, that there's been a lot of work with WDVAX and other WDVAX, where they've used cancer cells with all the antigens, so intact cancer cells with all the antigens that come with them, and that has not been led to safety problems to date, but it always is a potential concern, and it's one of the rationales for why going to defined antigens like neoantigens is really appealing. Because one, not only we have a much more concentrated antigen supply, but then you're getting rid of all the things that you actually don't want to break tolerance to. So certainly at this point still, I think it's safe to say still more of a theoretical concern, but I think a very legitimate one.
Okay, and then another question from the audience, and this is similar to one of the previous ones, but how does this vaccine impact the T helper cells?
Oh yeah. So we actually, it depends on how we formulate it, but we've actually been exploring the last couple of years this system actually for generation of humoral responses, which is not, I haven't talked about it all today. And we actually get a really significant impact on basically the helper cells. We get follicular helper cells expansion quite dramatically in the draining lymph node. We get actually a lot of activity in terms of increased CD4 numbers. We see then also impact in terms of germinal center formation and extended germinal center activity. So we are impacting that arm of the immune system quite a bit as well, even though I didn't talk about it, and probably at least some of the effects I described today are actually more coming, or at least in part coming functionally from that arm of the immune system as versus the CTL arm.
Okay, very cool. Okay. And so I believe that's all the questions that have been submitted by the audience members. And oh, Dan, we can't hear you if you're.
Hey, thanks very much for a great talk, a great presentation. I have two stupid questions for you. You mentioned in your presentation that two of the challenges you're facing are the cost and the time. So how do you make it faster and cheaper? Is it scalable? I mean, what are we trying to do on that front?
Yeah, so that's one of the things that we're emphasizing, where we think the materials can actually aid quite a bit. So both for the vaccines, we've been trying to develop vaccines that can be more rapidly deployed in patients, so we don't have to personalize them for each patient and then wait to manufacture until we actually have the patient, have the antigen identified. So if we can have a more universal vaccine, then that would have a dramatic impact in terms of time and in terms of cost. So that's really where we're trying to push that whole strategy. And so this antigen-less vaccine is, it's a really big rationale for that. And then for the CAR T cells, similarly, what we're hoping is if we can have a more rapid expansion and more functional cells, then you potentially could decrease the number of cells that would be needed for therapy, which would have a significant impact both timing-wise as well as cost-wise. So those are two things, scientifically, those aren't the most sexy things to drive your research, but practically they can be quite important.
Thank you again, and good luck with your research.
Thank you. Yep. Okay. Yep. Thank you for the opportunity today. Okay.
Hey, thanks again, folks. Stephanie, thank you very much for moderating. I do have a couple of quick housekeeping announcements before we go, if I may. Bear with me for just a moment. My slides don't seem to be advancing either, so I'll just do them from the application window. Are you seeing the application window, I hope?
Yep. Small.
Okay.
Yep.
Okay. Sorry about that. So we do have several additional upcoming webinars. 3D printing and orthodontics, restorative and regenerative dentistry. coming up on July 29th. And then in the middle of August, Synthetic Bio, jointly sponsored by the MRS. We're also soliciting ideas for new webinars. So if you have or know a colleague that you'd like to see present a webinar, please let us know. Send me an email to info@biomaterials.org. Ignore that typo. Same typo in my last. There we go. Also, just want to let you know we have received all of the ideas for Chicago, and we're sending out the formal call for proposals very shortly. And we'll be launching the abstract submission portal sometime around Labor Day with a abstract submission deadline at the beginning of November. Also keep in mind we have next year's joint symposium with the Japanese Society honoring 4 pioneers from the biomaterials field, 2 from Japan and 2 from the US. And that'll be at the end of July in Honolulu. And last but not least, because this webinar is open to all, if you're not yet a member of the society, please join. If you are a member, please get your colleagues to join. That's all I have for today. Dave, thank you again. Stephanie, thank you very much again. Have a great day.
Okay. Bye, everybody.
Bye.